So our water transport in plants lab is going to work like this, okay? In the lab across the hall, I've got some plants over there. We're going to take them apart and we're going to set up this lab to investigate what forces are present to transport water in plants. Okay, now we've talked about this in class already. Okay, we know that the force that drives water through the plant is an osmotic pulling action from above. As water evaporates from the leaf, salt gets left behind. Okay, that creates an osmotic pressure, and water is pulled from below in order to balance the salt concentrations in the spongy layer of the leaf. Okay, we've talked about that already, but what we want to do is investigate whether that's the only force and whether it can do it on its own. Okay? Because there's lots of people out there who say, well, if it wasn't for the roots, the plant wouldn't be able to transport water. Well, if it wasn't for the roots, the plant wouldn't be able to absorb water. That's for sure true. Okay, but the roots, other than the absorption of water, don't have a role in moving the water to the top of the plant. Okay, so what we want to do is essentially modify the plant a little bit so that only the leaves can act as the source of pulling force to move water. So the way we're going to do that is we're going to cut the leaves off. Okay, we're going to eliminate the roots as a source of force to move water. If the roots aren't there and the plant can still move water without them, then we know that we are right about where the force that transports water comes from. It comes from an osmotic pulling action in the spongy layer of the leaf. Everyone kind of follow me there? All right, so that's what we want to investigate with this lab. All right, so our variables. Okay, we kind of talked about one of them already. Okay, we're doing something to the plant. Okay, we're modifying it somehow in order to make sure that the only place that can be moving water is the leaf. Okay, you need to explain how exactly we're manipulating the plant in order to accomplish that. Now, what we're going to do in order to see if this water actually moves through the plant is we're going to color it. Okay, we're going to put food coloring in the water. The food coloring will move with the water, okay, as it's transported through the plant. And we're going to also measure how much water we put in, and then at the end of the experiment, measure how much water is left and see if any water has in fact moved. Okay, so we'll have two things that we'll be watching for change throughout the lab: the volume of water and the appearance of the plant. Okay, that is, where is the dye showing up in the leaf? Okay, and of course, anything that we watch for and measure is usually called a responding variable. Okay, remember again, we need to explain all these things. Now, for our controlled variables, any factors that could affect the plant's ability to transport water should probably be controlled throughout the lab. Okay, so you need to think of some things that are probably environmental that would affect a plant's ability to move water. Okay, so to give you some idea, the setup of the lab will be that we'll have these plants sitting in the windowsill in the chem lab for the next 2 days. Okay. The chem lab's temperature is fairly regulated. Okay. It doesn't fluctuate more than a degree or two. Okay. The window, they'll be right in the window. Okay. So over the next 2 days, the amount of light that they receive should probably remain somewhat the same. Okay. And of course, there may be some other things that might stay the same throughout the lab, okay, over the next 2 days. Okay, questions so far? All right, for our hypothesis, okay, since we are investigating the forces that move water in the plant, the if part should talk about what we think those forces are and where they're located. All right, so the if part says, here's what I think causes water to move in a plant. And that was my whole opening spiel here, okay, a few minutes ago about how a plant transports water. That's what should be worked in to the if part, okay, of our hypothesis. Okay? The "and" part of our hypothesis is just going to give a brief description of the experiment. Okay? We are eliminating the roots of these plants. We're placing them in colored water, and we're recording their appearance as well as the volume of water over 2 days. Okay, so if a plant transports water in the way we described, the "then" part should say what's going to happen over the next 2 days. Okay, that should include what's the plant going to look like, and what's going to happen to the volume of water in our container. Okay, so we have to be very careful in this lab about following the instructions for setting up our plant apparatus correctly. If we don't follow the instructions correctly, then the lab will not work. Okay, so we have to follow, and I know a lot of these steps are going to seem like really picky, but if we don't do them, it just doesn't work. All right, if we do them, it works flawlessly. Okay, but if we're not quite on the ball, okay, then our plant will not transport water and we will not get good data. So our experimental apparatus is going to look, in the end, like this. Okay, we will have 2 leaves, most likely, from the plant. Hopefully they're attached to the same piece of stem, but if not, that's not a big deal. Okay, placed into a small 25 milliliter graduated cylinder. Okay, and the graduated cylinder will be filled with colored water to very close to the top. Not all the way to the top, because if you fill it all the way and then put the plant in, what will happen? Water will spill out, and then we won't know how much of it is spilled out. We won't have an accurate volume reading, blah, blah, blah. Okay. Now, one of the things we need to also be aware of is that we want, as much as possible, to have the only place water can escape the cylinder to be the leaf. Right? We don't want to lose it to anywhere else. So we have to create at least a half-decent seal against the cylinder and the plant. So in order to accomplish that, I'm going to give you the finger off of a rubber glove. And you are going to use that to create a tight seal against the top of the graduated cylinder. Not all of you got that, but it's funny if you did. Okay, so we're going to use that as the seal. Okay, all you're going to have to do is poke a couple of small holes in it with a pencil and then stick the plant through those holes, or that hole if it's only one. Okay, then that will create a pretty good seal against the plant. We won't lose very much water out the top of the container. It'll only go out through the plant's leaves. Everybody kind of with me there? Okay. All right, so when we're setting this thing up, and it says this in your stuff there, okay, one of the things we want to do is make sure that the water is sufficiently colored that it will color the leaf, but not so colored that it's like a syrup of food coloring. Okay, so you're going to mix exactly 100 milliliters of water. So in order to do this, you actually need 2 graduated cylinders, right? You're going to need the small one that you're going to put your plant in, and you're going to need a 100 milliliter graduated cylinder to do the mixing. Okay, so you'll start off by coming up to the front with the 100 ml cylinder and putting 3 or 4 drops of food coloring in it. Right, now you have some options for colors. Okay, I have red food coloring and I have blue food coloring at the front. I used to put yellow and green up there, but as funny as it was when people used those, it just became tragic 2 days later when they couldn't tell whether any water had moved through their plant. So I don't put the green and yellow up there anymore. Okay, I only have red and blue. If you want to mix red and blue, that's fine. Purple is also pretty. Okay, it's up to you. 4 drops of food coloring in the 100 ml graduated cylinder. Then you'll go back to your lab station and put in enough water to fill it to exactly the 100 milliliter mark. Now, in using a graduated cylinder, okay, because water is a polar molecule, okay, it will cling to the sides of the graduated cylinder. And so when you look at it, it's going to make a shape like this. This is called a meniscus. Okay, and it's because, again, water is a polar molecule, so it's adhering to the glass. Our measurement is taken from the bottom of the meniscus. All right, so the bottom of the meniscus should be at exactly the 100 milliliter line. You need to get down and level with the graduated cylinder when you make that measurement, right? Because our plants are not going to move that much water over 2 days. If we are out by a milliliter or two, our data is going to be really messed up. Okay, so we need to make sure we're very accurate with our measurements. Okay, we're going to take that 100 mils of colored water that we've just mixed, and we're going to pour it into the small cylinder. Okay, and we're going to fill that cylinder fairly full, like above the end of the measurements and close to the top. All right, so you could see in this diagram here that, okay, I've got the water pretty much to the top. Not quite, because as we said, when we put the plant in, we don't want it to spill out. So very close. Let's say within, less than a centimeter of the top. Okay, we'll put that in there, and the top of the measurements is actually right around here. So this is where 25 mils is, but I'm going to add more than that because I need to. Right? This is why we're using the other graduated cylinder. I can actually measure more than 25 in the big one. Everyone kind of follow me on that? Okay, so I'm going to pour into there until, let's say, the water level is about there. Okay, so within a centimeter or so of the top. Okay, everybody with me? Yeah? All right. Then I'm going to look at my 100 milliliter cylinder and see how much is left in there. I'm going to go 100 minus that number. Okay, that will give me my initial volume of water. And that is a number that you will need to record as part of your data. Okay, so your initial volume of water is going to be 100 mils, okay, minus whatever is left. What has an H in it? Okay. Okay, that'll be your initial volume of water. Okay, it should be, around like 36, 37, something like that. Okay, all right. So once we've got the cylinder prepared, then we need to get our piece of plant. Okay, this is crucial that we do this right. Okay, this diagram is showing somebody doing it wrong. Okay, you can see that the piece of plant they've cut has 2 nice leaves on it. The problem is the end of the stem is out of the water. Okay, at the front of the room in the teacher's sink, I have the teacher's sink full of water, and I have the plants in the water. When you cut off your leaves, you need to cut them under water. Okay, cut them under water and put your thumb over the cut end that you are going to take back to your station. Okay, hold your thumb over the cut end, okay, and as best you can, after, of course, you've put the finger on there, okay, you're going to take your thumb off and quickly put the stem through and back into the water. Okay, we want to limit the amount of time the end of the plant is exposed to the air, because the longer it's exposed to the air, the more air will get into the xylem, and that disrupts the polar nature of the water that is in there. It can interfere with the hydrogen bonds and result in the plant not being able to transport water as well. Okay, so we want to have that end covered. Okay, bring it over, quickly take your thumb off, put it in. Now, in the picture here that I showed you earlier, look how far into the water the stem goes. Okay, it goes quite a ways into the water, and that is what you want. Okay, if you just cut off a little bit of plant and you only put it in this far, it's possible that enough water will evaporate out of there that the bottom of your plant won't be in the water by tomorrow. Okay, how well will this experiment work after that? It won't work at all. So make sure it's in the water fairly far. Okay, enough that it doesn't spill out, okay, but enough that it's also going to be in the water for 2 days. All right, so you're going to cut it under the water. Okay, and then, like we said, hold it over. Okay, and that's of course after you've put the glove on your graduated cylinder, poked a hole in it so it's all ready to go. The last thing you do is cut the plant and bring it over and put it in. Okay, everybody with me on that? All right, so when you have it all set up, okay, I'll actually come around at some point and just put some masking tape on your lab station. All right, write your names of your group members on that piece of masking tape because plants will all end up looking the same in a couple of days. All right, so put your names on that, wrap the tape around the bottom of the glove, okay, to provide a little extra seal on the bottom. You can see this one that hasn't been done on, okay, but you'll wrap the tape kind of around here and you'll have like a little flag that has, kind of sticking off of there. Right? That way you can identify your plant each day we look at it. Okay. All right. That's like as far as we'll get today is just getting it set up. We need it to run for a few days in order to see any change in the volume of the water. All right, we will talk about the analysis and stuff right now, but obviously we can't do any of that yet. Okay, we need to have data in order to do that. Now, the data we need to collect is as follows: the initial volume, and in 2 days' time, the final volume. Now, in order to get that final volume, I do have to pour whatever water remains in the 25 ml cylinder back into the 100 ml cylinder and just record the value. Okay, so that's how we'll do that in 2 days' time. The other thing you're going to want to record is you want to take a picture of your plant today and tomorrow and Thursday. Okay, we're going to take a picture of it each day so we can kind of show the progression of the dye traveling into the leaves. Okay, so we'll have 3 pictures: initial, midway, and final. Okay, those will also be part of your observations, and you can refer to them in your analysis, saying, on day 1, the dye was here, and on day 2, it was here, and so on. All right, so those will be all the data and observations that we'll have for this lab. All right, for the analysis and interpretation, question 1 is just calculating the amount of water taken up by the plant. That's a very easy calculation. Initial volume minus final volume. The difference is how much water was transported through the plant. Okay. Explain any differences between the initial and final values for volume of water, and we don't have to worry about this. In fact, I think I took it off of yours. We're not taking the mass of the setup. Okay. You'll also want to compare what you got with other people in the class. Okay, and then explain any differences. Okay, things you'd probably want to look at would be, how big were the leaves that group used, and stuff like that. Okay. Question number 2, what evidence do you have that water followed the path discussed in class? Okay, in class we discussed that water travels through the xylem, okay, into the veins of the leaf, where it then goes into the spongy layer and then is evaporated out through the stomates. That's the path we discussed in class. You need to show and talk about any evidence that you gathered during this lab to show that that is true. Okay, I would say your pictures are probably your most important piece of evidence for this question. And number 3, if we did this experiment again, but we used salt water instead of fresh water, what would happen? Explain why. Okay, so this is going to have to do with, okay, can you think outside the box a little bit in terms of cell transport processes and things like that? All right, then your conclusion, same as always, restate your hypothesis, explain why you accept or reject it based on the data and observations you made, and then sources of error. Okay, what are some sources of error that occurred, and how could we fix them in a future experiment? All right, any questions before we go across the hall to the lab? Okay, leave the Chromebooks here. All right, we'll go across, get everything set up, and then come back and have a little bit more time to work on that.